Good morning, everyone. Thank you so much for once again coming out to see the last presentation in our current speaker series. I'm happy to see the attendance growing and many repeat visitors. For the new folks here, I'm Brad Baker here on behalf of the Ontario Bat Network. The Ontario Bat Network was formed after the second Ontario Bat meeting was hosted by the Toronto Zoo in 2020. Following that meeting, we came together with the intention of ensuring that a third Ontario Bat meeting happened the following March. Well, March is well behind us now, so we made a compromise with the COVIDs and put on this virtual speaker series with the agreement that we could try again for the third Ontario Bat meeting in 2021. We hope that you've all found the speaker series beneficial and we can continue to get current research and bat work out to the broader Ontario community. We've got Gord Parker here. He's also part of the Ontario Bat Work Network to introduce our presenter, Craig Willis.
Good afternoon or good morning, wherever you are. Allow me to introduce Dr. Craig Willis. He is a professor of biology and chair of the Masters of Science program in Bioscience, Technology, and Public Policy at the University of Winnipeg. He and his students work on the ecology, physiology, and conservation of small wild mammals, especially bats, and have been contributing to the white nose syndrome research since the disease was discovered in 2007. Their work has been important for understanding the origin and physiological effects of white nose syndrome and the understanding of how we might protect bat populations from white nose syndrome impacts. Thanks for being with us today, Craig. Looking forward to hearing what you have to say today.
Thanks a lot, Gord, and thanks to all of the sort of network organizers for inviting me to be with you today. Yeah, we've got sort of mixed feelings. I'm talking to you from Winnipeg. We've got mixed feelings here in Winnipeg at the moment. We're very excited that the Jets swept the Oilers over the last week, 10 days. And sorry, Ontarians, we're looking forward to crushing either the Leafs or the Canadians in round two. So we're happy about that. We're a bit bummed to be the COVID capital of North America at the moment. And so that's a bit distressing. So it's nice to be able to talk about a non-COVID disease of bat origin or likely bat origin, our fungal pathogen that we've been working on for the last 10 years or so. I want to try and tackle three questions that we've been interested in the last decade or so, my students and I. And I know from seeing some of the names in the participants list, a bit of this will be review for many of you. You will have seen some of this before. Some of it will be new, I promise. But yeah, we'll have some review material of stuff you've probably seen in the past. So one of the first things we were sort of interested in is how white nose syndrome affects the physiology of individual bats. We know it's killing bats in huge numbers, and we'll talk more about that in a second. A second question we've been trying to address more recently is how white nose syndrome might change populations. So are individuals changing their behavior, or are populations evolving? I'm not going to be able to tease out those two factors in the data I'm going to show you today, but we're interested in how those changes might be occurring and what might underlie them. And finally, what they might mean for what we should do to help protect and recover remnant bat populations after white nose syndrome invasion. Want to, of course, acknowledge a huge number of students and postdocs and amazing collaborators that have actually done the work I'm going to talk about today. Definitely don't have time to mention all of them by name, but certainly want to highlight that this is very much a team effort. And want to leave you with a couple of sort of take-home messages right from the beginning. One conclusion that I think we already know before seeing this talk or is in the literature long before our work is that white nose syndrome is in fact changing the biology of impacted bat species. There are now a couple of really nice papers showing evidence of evolutionary change in the genomes of remnant bats left after this massive bottleneck has gone through and killed lots of bats. And we'll touch on maybe a couple of those genetic changes a bit later and how they might relate to the phenotypic or the actual real world trait results that I'm going to touch on. Also want to highlight variation. There's very likely not a single silver bullet cure for this conservation problem. Like so many conservation problems, it's complicated. And what we do in one place for one population might not necessarily be the best thing to do in another place for a different population. We're going to need a toolbox, a toolkit of multiple strategies to try and help bat populations persist and recover. So a little sort of history of my group's involvement in white nose syndrome. We sort of first became interested and involved around the time these early die-offs of bats were being found by biologists in New York State, going into their annual surveys of bat colonies and instead finding carpets of corpses, little skeletons, and a few persisting survivors with this white stuff growing all over them. This was happening during hibernation, and my students and I were interested in the hibernation biology of bats. And so sort of by default, we ended up involved in trying to understand what was going on. Around the time, very soon after, in fact, these die-offs were first observed, some really great biologists from the US had worked out that this white stuff growing on the bats was this fungus, now called Pseudogymnoascus destructans, or PD, that it was cold tolerant, so it grows very nicely at the body temperatures of hibernating bats, but certainly not at a normal or warm mammal body temperature. And finally, that it was only invading the skin and causing a cutaneous infection. And so quickly, a science strategy meeting was convened in Albany, New York. And I was asked to go because I worked on hibernation in bats. But this was a meeting of wildlife disease experts and pathologists, which I certainly am not, people that knew a lot about diseases of wild animals. And one of the sort of persistent comments I heard frequently throughout this meeting was that fungi don't kill mammals. They don't kill mammals and birds. They really like plants because we think plants tend to be cold or colder than warm-bodied mammals and birds. This led to sort of the hypothesis that the fungus was some secondary problem, that there was a virus or pesticide exposure or the wrong kinds of food or not enough food in the fall that was causing bats to be in rough shape. And this white stuff growing on them was an opportunistic fungal infection. A few of us that studied hibernation, though, wondered if maybe because hibernating mammals, especially bats, with their really long hibernation times and very small energy reserve, they're very sensitive to disturbance. And so we wondered if maybe the simple cutaneous infection could really be causing problems for that hibernation energy budget. And so to address that, we tried to understand how this simple cutaneous infection might change the physiology of bats. To tackle that, we first did inoculation experiments where we took healthy bats from the wild. We outfitted them with little temperature backpacks that record their body temperature so we can understand hibernation. And then we either inoculated them with fungus or sham inoculated them with a control solution and then allowed them to hibernate in natural conditions. And we saw a few things. We saw bats that were inoculated with nasty looking wings and lesions. All of this squiggly stuff is fungal hyphae that's breaking open the skin of those inoculated bats compared to the nice intact wing membrane of control bats. This is a cross section through that wing tissue and a cross section through a white nose syndrome lesion. That skin infection just looks nasty at this magnification. And it turns out it does mess with the physiology of bats in ways that affect their hibernation. And so this is a graph that I'm sure many of you have seen a bunch of times. It's effectively showing what all hibernating mammals do during hibernation. This control trace is the skin temperature of one of our healthy sham inoculated bats. These traces look exactly the same for free-ranging bats in the wild. Basically, they spend most of their time at this very low body temperature, which saves a ton of energy. And then every few weeks, they warm up to a normal body temperature. We're not totally sure what these arousals are for. They probably serve a bunch of different functions, but whatever they do, they cost a lot of energy. Each of these little spikes in body temperature costs about 3 weeks of cold time. So they only warm up for a couple of hours, but they burn through a ton of fat to do it. Our control bats look like free-ranging bats, healthy bats. Our inoculated bats warmed up about 3 times too often by the end of hibernation. And so if each of these little spikes equals 3 weeks of torpor or cold time, that's a big expenditure of fat that these animals can't afford. Why would bats with a simple cutaneous skin infection warm up too much? Why would they burn through their fat so quickly? Well, one of the questions we asked was why healthy hibernators arouse in the first place. And some smart folks, especially Paul Cryan and his colleagues, came up with the idea that, that perhaps dehydration is driving this increase in arousals. We know that bats can lose a lot of water across this delicate, highly vascularized wing membrane. And one thing that can cause them to warm up from torpor during hibernation is increased rates of water loss, being in a dry environment. Perhaps the fungus was causing this to happen to the wings if those lesions are breaking open the skin. And so we looked at that in a couple of ways. When we looked at the blood of our infected bats, they had really low electrolyte levels. So that suggests maybe they're losing fluid. We also used a technique called open flow respirometry to actually directly measure water loss in infected and control bats. So we used all of this complex doohickey machinery. We housed bats that were either infected or sham inoculated in these little respirometry chambers. And then we pump air into those chambers that has a known quantity of oxygen and carbon dioxide and water vapor. And we can measure how much water vapor bats add to the airstream when it comes out of that chamber. And what we found is that both in both humid air that kind of matches what bats might hibernate in, and in dry air where we'd removed all the water first, or most of it first before it goes into the chamber, our infected bats had dramatically higher rates of water loss than controls. And importantly, that level of evaporative water loss for the infected bats was correlated with how badly damaged their wings were due to white nose syndrome. So a clear link between fluid loss, water loss, and ultimately this increased metabolism during torpor and this increased frequency of arousals. Essentially what's happening, the physiological change that's happening is this skin infection is catastrophically messing with the hibernation energy budget of badly impacted bat species. And as a result, they starve to death. Turns out they starve to death, or we think they starve to death. There are other actually physiological consequences that could also kill them, but they're certainly burning through their fat reserves way too quickly. Where are we now in terms of the sort of community impacts for bat communities across North America? We've got about 20 species that have been found to have the fungus on them. 12 of those have been more or less affected, some of them dramatically so. Now, and we also know that this fungus survives quite nicely in the environment, even without bats. That's a particular worry. That's shown really nicely in this outstanding paper by Fred Frick and her colleagues, In this graph for a particular cave, we're looking at here basically the load of fungus in the cave and the population size or the group size, colony size of the bats. And you can see that this fungal load increases from its first detection, sort of concomitant with the crash in the bat population. And then you've got this tiny bat population down here. The fact that this fungus persists so nicely without bats, and the fact that you've got multiple species, not all of which die from the disease, means you can have horrible, horrible population impacts. Transmission keeps happening, even if hosts are relatively rare. And these kinds of pathogens can drive hosts to extinction, right? So we've seen millions of animals killed. We've listed 3 species as endangered in Canada. One has been listed as threatened in the US so far, entirely because of white nose syndrome. And it's spread incredibly fast. I'm sure most folks have seen the maps over time. We start back in 2007-ish with this red X. And in the sort of 14 years since that time, since it first was detected, we're now looking at it having spread all across the continent. Okay, so we've seen important changes in the physiology of individual bats, and we've seen an obvious change in populations. There are simply less bats of the highly impacted species. It's caused their populations to absolutely crash. The second question, though, that we've been interested in, and lots of folks are interested in, is what have those crashes meant for the characteristics of our remnant bat populations? One important feature of the disease, I've shown you these data from Fred's paper for a couple of hibernation sites. This isn't the pattern everywhere. In some places, bats are persisting and even recovering. So we're seeing recoveries in some places, which suggests the possibility that some characteristics of these bats, of these survivors back here, were important for resisting or tolerating the infection, important for surviving the disease. And that raises the possibility that bat populations are evolving in response to this massive selective bottleneck. A really interesting question and an important one for conservation, because if they are evolving, we might do different things than if they seem like they're just going to be driven to extinction. Right? One of the things that we've highlighted so far is that that energy reserves during the winter should be important because of this increased arousal frequency and increased energy expenditure. And so that led Tina Chang and her colleagues, and I had the good fortune to be involved in this study, to test something that we called the fat bat hypothesis, that perhaps how fat a bat gets in the fall before hibernation predicts whether it might survive and persist after the disease. This is an important question because we know, first of all, that white nose affects energy expenditure during the winter. We also know that how fat hibernators get before hibernation is a heritable trait, at least in some species. We don't know for bats whether it's heritable, but in some ground squirrel species, we know that it is. They can pass this on to their offspring. And so if this trait is heritable and there's a massive selective pressure caused by white nose syndrome, we should see fall fatness become more common in our population after that selective event. And that's exactly what Tina and the rest of us found for at least some colonies, some hibernation sites in the northeastern US. What we're looking at here are data for body mass. We actually measured for this study fat content of the bats using a technique called quantitative magnetic resonance, but it doesn't really matter for the story because fat mass and body mass are so tightly correlated for bats, especially at this time of year. So what we're at here are measurements that were taken the fall right before the crash from white nose syndrome happened. That's what this line is showing here. Population crash. This is how fat bats were right before that crash. We went back several years later, measured how much fat or how heavy bats were at that same time of year right before hibernation. And what you see is a dramatic increase, a significant increase in how fat bats were. This plot over here is simply showing the colony size in this particular cave where we happen to have a good count, right? So this is definitely happening in some places. If I zoom out from these two panels of this graph to this graph, we can see that it's, here's the two panels I just showed you. Here are some others where that's also seems to be happening. Bats are getting fatter after the crash, but there are a couple places where it doesn't look so simple, right? In a couple of these sites, different caves, the bats aren't any fatter several years after the population crash. This suggests the possibility, certainly doesn't confirm it, but it raises the possibility that this isn't the only thing likely to be different in post-white nose syndrome populations. Other things could be selected for. And of course, there are a whole bunch of possibilities. We wondered about this population difference, and we also wanted to look at a northern population in terms of this fat bat hypothesis. So we came back home to Manitoba and studied a population of bats that has to hibernate in some of the longest, coldest winters that hibernating bats experience anywhere, where our little brown bats routinely hibernate for 8 months out of the year, from September until even early June. So they are marathoners of hibernation. They really have to tolerate a long period of hibernation. What could that mean for this fat bat hypothesis? We caught bats during fall swarm. We'd already been collecting data before white nose syndrome arrived, and we compared our pre-white nose syndrome data to post-white nose syndrome data. What did we find? Well, no significant effect in the hundreds of individuals we were able to measure. There's no detectable increase after the population crash, despite a big population crash. What's interesting here is how fat our bats were, though, already. If I show you the sort of body mass of Tina's, even post-white nose syndrome bats, her bats remaining after white nose syndrome went through and presumably selected for the bats that got the fattest in the fall, they're still much smaller before hibernation than our bats. These Manitoba bats are already accumulating a huge fat reserve before hibernation. It could be that they were just already maxed out. They have no capability of getting fatter, or at least dramatically fatter enough to make a difference. Again, suggests that regional patterns of change in our population may occur. We might see differences and different patterns in different places. Also need to highlight that for both Tina's study and this one, we don't have good data on the heritability of this trait or the other traits I'm talking about. So we could be talking about evolution if we're talking about changes in trait values. We could be talking about within individual flexibility of survivors. We don't know for sure. Okay, what are some other traits we could look at? Well, there are a whole bunch. There are things that happen during hibernation. There are things that might happen during spring recovery from the disease for survivors that could be important. There are immune system traits that might be important, and folks are sort of actively investigating all of those things. One of the traits that my students and I have been looking at, largely out of convenience, because we happen to be measuring it before white nose syndrome emerged in our study system, is so-called animal personality. Personality is maybe not the best word. Temperament I always liked better, but I wasn't consulted, and it's now a standard term in behavioral ecology. Consistent individual patterns of behavior or consistent individual differences in behavior, like sociability, the tendency to hang out with other individuals, or exploration, how inquisitive, how curious individuals are about their environment, We all know people that vary in these kinds of traits, and it turns out that individual animals vary consistently in these types of traits, and this can have big implications for ecology, right? The most sort of commonly used example of personality refers to the sort of shy, bold continuum, where you've got sort of passive, quiet, maybe introverted individuals at one end, and then bold, aggressive, explorative individuals at the other end. We see these continua in bats, in rodents, in fish, in all sorts of other animals, and also in people. They're really well quantified, and they can influence ecology. In bats, we know that these kinds of traits, measured using the standardized tests that we use, developed by people studying rodents, really, we've co-opted them for bats. We know that they're repeatable in bats. They're also heritable in many taxa, but again, we haven't worked out heritability of these traits in bats. We can't say for sure that they pass them on to their offspring, although it seems likely given how, given the heritabilities we see in other taxa. So in terms of bat personality, we've been studying personality traits of bats for a number of years to try and understand things like roost selection. How does behavioral performance in a standardized test correlate with an ecologically relevant pattern of behavior, like roost selection or social network dynamics? It turns out that those standardized tests can actually predict how individuals behave in a natural or semi-natural environment. And we also have found some evidence that they affect the transmission of pathogens and parasites. As we've all learned over the last year or so, much to our dismay, social distancing affects pathogen transmission, right? Turns out that's the case in terms of personality as well, that bats, which tend to be highly active and explore their environment and interact with lots of other individuals, can influence pathogen transmission in their environment, right? We've tested that experimentally, and we wondered whether sociability of different individuals might be a trait impacted by white nose syndrome, whether it might change in individuals. To measure sociability in bats, we can use a couple of tools, but one of the most common tools, again, developed from rodent studies conducted by psychologists, is to use a Y maze. So we do, say, a 10-minute standardized test that we video record. We have a stimulus bat in one arm of this Y maze and nothing, an empty cage in the other Y. And then we release an individual into the maze and we just video record its behaviors. How much time does it spend close to this new bat it hasn't met before? How much time does it spend in the other arm or back here hiding out from this new bat? That allows us, by quantifying a bunch of different behaviors, to calculate a sociability score. And when we do that, what we found was, a bit unexpectedly, when we compared bats that we'd been measuring before white nose syndrome, these are bats all from 1 year, 2014, where we measured sociability in the Y maze. After white nose went through the population at this particular site and caused a big crash, we actually see higher values of that sociability score. We kind of had predicted that sociability might go down because we thought sociability might be a disadvantage, as it might increase your risk of acquiring infections with this fungus, right? But in fact, it goes up, and we're not totally sure why that is. We think it's interesting. We also think it might be important. One of the reasons that we think this might be happening is mechanistic. One of the genes that's been implicated as under selection by white nose syndrome is a gene known as FOXP2, which Oteri and Knowles showed appeared to be under selection after a population crash in the population they studied. We don't know for sure what it might do in bats necessarily. We know it's important for speech in people, for acoustic communication, for vocalizations in rodents, and for echolocation in bats, it also appears to play a role in social behavior. So this gene, the fact this gene is also selected for by white nose syndrome, and we see these changes in sociability, suggests maybe a link. We certainly are nowhere near confirming that link, but it raises some interesting hypotheses. Why sort of ultimately, why might it be beneficial for post-white nose syndrome bats to be more sociable? Or why would white nose syndrome select for more sociable bats if that's indeed what we're seeing here? Well, one reason for that is that social thermoregulation could be especially important for white nose syndrome survivors. We already know that bats like to cuddle in the summertime, even before white nose syndrome, to help their pups grow quickly, right? Bats recovering from white nose syndrome in the spring are faced with two big challenges. Their wings basically start to fall apart as they come out of hibernation. They have this massive inflammatory response that causes all sorts of extra wing damage, and they then have to recover and heal. Healing requires you to be warm, to have a warm body temperature. If you're going to heal and reproduce, you've got to get warm, right? They face the second challenge that many of their former colony mates may be dead. They may have died from white nose syndrome the previous year. You're much less likely to be able to find your old pals from the previous year. And so it's possible that a greater tendency to be sociable could be really important in the spring for allowing bats to seek out new colony mates to effectively cuddle with, to do social thermoregulation with, so that those individuals can heal more quickly, can recover from white nose syndrome, and potentially reproduce, survive and reproduce that summer. Okay. Again, zero evidence for this. This is speculation. What we thought was interesting was this increase in social tendency or sociability. Okay, so some ideas and a bit of data about how white nose syndrome might change populations. I want to shift gears for the last part of the talk and sort of address some ideas about what we might do to help bat populations, help remnant populations persist and recover. Well, one of the first things that happened once it was confirmed that the fungus was really causing all this mortality and these die-offs was that people started cleaning their stuff. And that helped really dramatically reduce human spread of the fungus. This was an important early decision. It's obviously hasn't come anywhere close to solving the problem, but it's helped us avoid making it a whole lot worse. One of the other things that happened fairly early in the progression of the disease across North America, we were probably a little sooner than this map when the decision started to be made that we needed to list species. Their populations were declining fast enough that they needed to be afforded special protection. In Canada, this decision kind of flew under the radar a little bit. It wasn't that controversial, and 3 species were listed as endangered relatively quickly and relatively smoothly. But in the US, this was a bit of a controversial decision because of the automatic habitat protections that come along with listing. And so you had statements in the media from this politician from the US that you probably never heard of. This was a time in the US when actual policy seemed to be discussed a whole lot more than other issues. Here, this Republican from Utah, who was chair of the House Natural Resources Committee, was quoted as saying, this decision to suggest we should list a bat species as endangered or threatened will fail to solve the real problem, will fail to mitigate the real menace to this species, which is a disease called white nose syndrome, not human activities. And so the key point in here is don't mess with industry, don't provide this automatic habitat protection, and instead cure the disease that's actually killing the bats, right? This led to what, in my view, was a bit of an imbalance in the kind of research that started happening and that was being funded. a lot of which is incredibly promising and a lot of which can be really useful as part of a management solution and as sort of an adaptive management approach. But this emphasis on curing the disease as opposed to providing habitat protection, including summer habitat protection, had me a bit worried. And for a while, I got a bit down on actually trying to treat white nose syndrome to the point I made up an acronym. Spray on bats is what it means. It doesn't mean anything else. Many SOB treatments were developed and tested in both the lab, and now some of those are being tested in the field. Some of them show a lot of promise. Some of them not so much. My group has tested some of them as well, and we've had some in the positive and the ineffective camp, that's for sure. I think what concerned me though at the time was this imbalance, this sort of teeter-totter swinging away from protecting habitat. One of the first, this isn't really a spray on bats approach, it's a sort of provide to bats approach. One of the first things we tested, because we knew bats were starving and we knew they were low in electrolytes, was to use Pedialyte, which we use to treat dehydrated people, but we also use to treat dehydrated wildlife. If there are any wildlife rehabilitation folks on the Zoom, what we did, this is a view looking down from the top of one of our cages in our artificial hibernaculum in the lab, and here's a cluster of bats. One individual aroused. We placed multiple dishes on the floor of the cage and allowed bats to choose a 50% Pedialyte solution, a dextrose solution without electrolytes, and just plain water. And what we found was absolutely no benefit in terms of survival. And no benefit, not surprisingly, there wasn't a benefit in terms of fungal load. This didn't help bats fight off the fungus, and it didn't help them do any better. So an example of an ineffective potential treatment. The reason we tried this to get bats some energy is because we know they'll drink in a cave during hibernation, but they have to be trained to eat. You have to train them to eat mealworms, as most of you probably know, and that's unlikely to be successful in the middle of hibernation, right? But if we could have gotten them to drink this stuff, we could have maybe put a trough of Pedialyte in a cave, and perhaps that would help bats survive. Results of this experiment suggest unlikely. There are lots of other things that might help reduce the load of fungus on bats or in caves. One of them that's showing a bit of promise lately is UV light. And one of the reasons it's appealing is that it seems to be pretty specific to PD in terms of the microbes it kills in caves, because PD is a very cave adapted microbe from what I understand. And so these are just sort of growth plates of fungus being treated with various levels of UV light. I think there are a bunch of studies in progress. We've tried one where we tried to decontaminate PD from cells in a cave, compared it to a couple of other potential treatments, and didn't find a really sort of staggering benefit of that treatment, but does show some promise from what I understand in other studies. Another thing we tried was a bacterium that was isolated from the wings of big brown bats, a resistant species, and it's shown here in this dish happily inhibiting the growth of bee. This strain of Pseudomonas fluorescens, we applied it to the wings of infected little brown bats, and we were able to see an improvement in survival of about 30%. And a subsequent field trial also found an improvement in survival, suggests some promise for this potential probiotic. Lots of other things in development, lots of other things being tested, but a couple of important criteria need to be satisfied. And this is one of the reasons that I think there's no magic bullet to solving white nose syndrome. There are going to be multiple strategies that will be required. First, the effectiveness of the treatment has to be very high. 30% improvement in survival might not be high enough to actually help your population recover. Right? And we need to be able to find bats to treat. And a lot of the spray on bats kind of treatments require finding bats in the winter. Throughout much of Western America, any clue where our bats are during the winter, even though we know there are lots of bats in the landscape in the summer, right? So they're there, we just can't find them. If we can't find them and access them, we can't get them the treatment, right? Again, some treatments have ways around this, but many don't. And so, again, unlikely to be one sort of silver bullet. And I also think it's important not to forget about habitat, and in particular, summer habitat, so that we can help remnant bats survive and reproduce. And if they possess heritable traits that help them survive, they pass on those traits to their offspring. One of the things we've known for a long time about bats is that they like it warm in the summertime. Obviously not overheating temperatures, but really very warm and relatively stable, right? This is one of the reasons they cuddle, and they often choose rooftops or sites in tall emergent trees that get lots and lots of warm sun to help their pups grow quickly. As we've mentioned, this is probably also going to be really important for helping recovery from infection in the spring. And so to test out this idea that really good quality summer roosting habitat could help bat populations recover from white nose syndrome, we built hot boxes for bats. Could we improve spring and summer roosts for white nose syndrome survivors? Basically took standard 3-chamber bat houses, we insulated them and outfitted them with a heating coil and a thermostat. We then covered them in sort of black tape and set the thermostats to 30 degrees, which is the low end of the thermal neutral zone, the temperature at which a mammal has to start shivering, basically to maintain body temperature. We set them to 30 degrees. They're open at the bottom, so they actually rarely exceed 30 degrees, only on a really hot day would they get that hot. But we set them that way, so the heating coil is usually trying to warm them up, especially at night. We did a bunch of tests in the lab, and bats liked them in the lab in captivity. They tended to use them really reliably, but we wanted to test them in the wild. And so we used the Neighborhood Bat Watch citizen science program that we helped expand from Quebec into Ontario and Manitoba a few years ago to identify colonies in Ontario and Manitoba where we had really keen participants that would upload their sites and upload count data for us, and were very keen about their bats, and would also agree to let us put experimental bat boxes adjacent to a little brown bat colony that was already there. Okay. This is just a map of our sites. You can see they're spread across a really big area. The yellow are our heated colonies. These colonies got a heated bat house. The dark circles are our control colonies that just got a bat house, no heated bat house. And all of these sites white nose positive by 2019. So we managed to get hot boxes at 19 colonies and control boxes at 20 of them by 2019. We placed our boxes at a maximum of 20 meters from the entrance to an existing colony to increase the chance our bats will find them. And in this data set, we also include 12 other control colonies in addition to our 20 that got control boxes. To give you a spoiler, the reason for that is because we got so few bats in these control boxes. It was very tough to make comparisons. So we added some other colonies that already had bats so we could get some measurements. And we think roost switching between any of our experimental colonies is unlikely. They were all at least a couple of kilometers apart, and most of them much further than that. So reduces the chance bats were switching from site to site. We visited each of these sites twice per summer. So once during pre-volancy before pups are flying in June, once after pups are flying in hopes of detecting a surge in new offspring that have emerged and are flying for the first time. And we measured a couple of things. We looked at adult reproductive ratios and numbers of juveniles and body mass and those kinds of things. We also placed drop sheets under the entrances of our roosts so we could quantify fecal cortisol, which is an index of chronic stress. Okay, and then we netted at roosts once each of these visits to as close to the roost as possible. We couldn't necessarily get right at the entrance, but we got as close as we could. So what did we find? Well, first of all, we found that our hot boxes were warmer. Here's our heated box line up here. Here's our unheated line, much closer to ambient temperature. And encouragingly, they also didn't overheat. So because of that insulation, we think they maintained a cooler temperature on extremely hot days. They were warmer and were stable as we'd hoped. What did we see in terms of the bats? Well, we found a significant reduction in fecal cortisol in our heated boxes or in colonies with heated boxes relative to control colonies. So a reduction in apparent chronic stress for bats that had access to a heated bat box. And we saw that during the prevalency period, pregnant females were heavier, and in the volancy period, we had a 3 times higher adult to juvenile ratio at colonies with heated boxes compared to control colonies. A whole lot more juveniles, a whole lot more reproduction at these heated colonies. So these results together suggest some promise. We're still collecting data. And one of the things we really want to rule out is that these heated boxes increase transmission of PD, right? We don't think that's a huge deal in the summertime, but it could be important. And so this is why we're certainly not yet at a stage where we're recommending people go out and buy heated bat boxes and set them up everywhere. We didn't really do this experiment to test heated bat boxes per se. We did it to test the potential of habitat improvement, summer habitat protection to help white nose syndrome affected bats in general. And I think it argues to us that we need to do some difficult and expensive work to understand how we can enhance natural roosting habitat for bats. There are certainly species that won't go in a bat house or are much less likely to go in a bat house. So the heated bat box is not, again, anything like a silver bullet, even if we can show it works really well and doesn't increase the spread of PD. But what these results suggest to us is that summer roosting habitat could be really important to protect and improve if we can. So just to summarize all that in the last minute or so, I think in terms of the 3 questions I set out to sort of address today, what our work's helped to show, and in combination with work from a lot of great research groups across North America, really we're looking at fluid loss from a simple skin infection leading to this catastrophic disruption of winter energy balance. We think we've got some hints about how white nose syndrome might be changing populations. And there are other hints about other traits out there. Remnant little brown bats, at least in some sites, have more fall fat. Not all sites. They also tend to be more sociable in at least one site. There are likely other traits, and it remains to be worked out whether these differences reflect evolution or within individual flexibility. In terms of what we should do, I think the sort of take home that I'm going to leave you with next is that probably a bunch of different stuff, and it depends on the population that you're trying to protect. But I think we can't rule out high quality summer roosting habitat as a way to help bat populations recover. So the sort of take home messages again, white nose syndrome, I think, is changing the biology of affected bat species. We know that from the genetics. We've got some interesting and I think potentially important hints from the phenotypic values that we've looked at and others are looking at. And I think there's not going to be one silver bullet for white nose syndrome. We're going to need to try lots of different things and have a full arsenal of management options we can try and use. And with that, I will thank you very much for your attention. Thank all of these folks, and hopefully I haven't missed too many, for all of their help and hard work. And if there's a minute or two, I'll try and answer some questions.
Awesome. Thank you so much, Dr. Willis, for your presentation today. There's a lot of information to process over the last hour, so it's excellent stuff, and hopefully we'll have some good questions for you from the participants. I have two questions written down here so far. And it's gone. There it is. Happy to see that there are multiple researchers that are showing populations from some species appear to be recovering. The fat bat hypothesis is interesting. I'm curious, we had another presenter speaking about bats with infections aside from white nose, which could become a problem due to the increased stress on the individuals. Do you know if there have been any researchers considering how that may reduce the potential that survivability, like fat bats or other characteristics, may increase in the populations?
It's a fantastic question. I think, you know, there's some interesting coronavirus results in that respect. So our myotis in North America have a commonly have a coronavirus. We can't get it. We don't call the newspaper, obviously, but when bats have white nose syndrome, they're clearly stressed, and a lot more of this coronavirus can reproduce, right? They have a lot more of the RNA of this coronavirus. There aren't good links to survival, though, I don't think. What we did find in that study, where in our lab bats, basically what happened in one of our early infection experiments, we brought back bats into the lab. About a third of them just happened to be infected with their myotis coronavirus. And in those bats, their white nose syndrome symptoms were more severe. So suggests that there is an interplay between pathogens that could be really important. But yeah, we don't have good hard links between co-infection and survival or mortality.
Excellent. Julia Sunga has a question here. So Julia, if you want to turn on your camera and mic and ask the question, that'd be great.
Sure. So you mentioned that you don't think these bats are switching between the roosts that are available, especially when you add those higher quality summer roosts. So do you have suggestions of how we could make bats maybe find those areas and switch to those higher quality areas, especially if, you know, I'm looking at social stuff, if they're losing their associates in the winters that they would normally be following to these areas?
Oh, you know, fantastic question. I think it's, as my father used to say, it's not a matter of money, it's only a matter of the amount, right? So these are super expensive questions to try and get at, I think. Some of it you can get at with population genetics. Like, we still don't know how the group of bats that's in a hibernaculum relates to the group of bats that's in a summer roost. My hunch is that you've got matriline's of bats moving together, and the hibernation colony splits up, and then you've got, you know, a group of females that all go together to the same summer roost. And a bunch of those different matrilines all convene. So you've got little pockets of sociality within each thing. Incredibly tough. We're trying some radiotelemetry this summer to better understand roost switching at some of our experimental colonies. I think that we know a fair bit about roost switching in the forest from tree to tree to tree. And people tend to assume that bats in buildings switch less, or bats in bat houses switch less, but they still switch, right? If you go and net at a colony, they all move the next day, or most of them move. So we're trying to understand backup roosts and how they move around. I think the key for understanding, for having at least a good argument to policymakers that certain habitat features are important for populations, is to somehow connect features we can measure with individual survival estimates, right? We look at preferences a lot. So bats like this kind of tree and they don't tend to use this kind of tree. But does that affect survival if they're forced to use the tree they don't necessarily choose? I think we need some really creative approaches and probably some technology that may not even exist yet to try and get at those kinds of questions. How do those preferences affect actual survival and reproduction? So that was a long-winded non-answer, Julia.
Great, thanks.
Thank you, Julia. I got another question from Elizabeth Clare.
Hi, Beth.
Hi, Craig. It's been a long time.
Yeah, nice to see you.
I have a naive, simple question. I've been out of the country and the continent and the world of white nose syndrome for a long time now. Whatever happened to the experiments cross-infecting European populations and trying to look at where the origin of white nose syndrome was? I don't think I ever heard the conclusion of those studies. Did they have a conclusion?
Yeah. So Christina Davies published some of the transcriptome data from those experiments. It's now clear. I don't think we're going to find out where exactly in it came from, but it's now crystal clear. It's of Eurasian origin and it's everywhere. We were doing an experiment where we went to great expense and effort to import European myotis and try and do a side-by-side experiment. And sadly, these experiments go sideways sometimes, and that one went sideways with incubator failures and those kinds of things. So the side-by-side experiments, getting to the point where we got in the arousal changes, those haven't really happened, but there's a couple of cool transcriptome studies that show differences in the responses of European bats and North American bats, clearly doing different things, responding in different ways, and comparing susceptible and resistant North American species too. So there's some stuff out there for sure.
Thank you.
Okay, thanks.
Another question from Elizabeth Bainan.
Hi, Dr. Willis.
Hi.
So my question is, oh wait, I can put my video on as well. Sorry. So my question surrounds sort of the recommendations that you'd make if you were to exclude a colony from a building or a built asset. I've heard some comments about maybe there being overheating in bat boxes. So, and when like the dehydration increases in infected bats, I'm kind of concerned, but I can't find any articles on that in Canada. So maybe it's not too much of a problem. Are there other measures you could kind of do, or is bat box the best?
Yeah, I think options. I think they like to have options. It gets back to the roost switching thing I was talking about before. If you give them multiple places to go, and usually they do have a backup. I think they're probably pretty good at predicting the weather too. So they probably don't go into the same spot on a blazing hot, sunny day. They choose in the morning before it gets blazing hot to go to maybe a shadier spot. No data on that, but that would be my hunch. But we also find them in Manitoba, we'll find them underneath the aluminum roofs of a shed or a barn where it's got to be over 40 degrees during the day and they're still in there, right? So I think folks in the States worry a lot about overheating, and probably Southern Ontario, it's going to be a big deal, but it depends where you are, I think. And I'd say options are the best. So if you can give them as many different places to go as you can. If you're excluding a big colony, put up 3 posts with 4 bat boxes on each one in different places. That would be my sort of high level kind of idea.
Perfect, thank you. So it's the variety of microclimates.
I think so. That's a hunch, but I don't, yeah.
Great, thank you.
Thank you for your question. All right, I don't see any others. I have one more if you don't have mine. I've got habitat protection has been a bit of a discussion in Ontario since the listing of species as well. Do you have any thoughts on how we could work to encourage protection or enhancement of important summer habitat features? And I mean, maybe it's not backed up by science yet, but maybe you have some thoughts because you deal with them a lot.
Yeah, absolutely. I think we're starting to get a handle on, well, I think little browns are going to persist, but long-eared bats may not. They may go extinct. So it's just like a researcher to say we need more research, but I think we need to understand their habitat requirements and what affects their survival a whole lot better if we can. That's a very tough and expensive proposition. Failing that, I think, the studies show that a mix of forests, probably lots of wetland. So, clear cutting and a new sort of replanted tree plantation, probably not going to be as valuable as a mix of forest types, forest stand ages, those kinds of things for that species. And I think in many ways we're used to thinking about losing bats as a problem because it means we're going to have more insects, more pest insects. And that may be true, but I'm starting to look at it more from the perspective of the insects, because we're here also seeing so many headlines about crashing insect populations all over the world. And little browns seem, thanks to work from Beth, and she can correct me if I get it wrong, but they seem to really like wetland insects. They certainly eat everything, but little browns in particular seem to eat a lot of wetland insects. So if we are conserving the wetlands that support those insects, I think and conserving them close to the roosts of bats, close to places where bats live to make it easy for them, I think that is likely a good strategy. In many ways, I think insect conservation is going to be. Okay, I'm not hearing Brad.
I said, thank you so much. I'm back here.
Yeah. Okay. Good.
Yeah, so it looks like we're having people signing out now. So I think we're at the end of the question period. Thank you so much, Dr. Willis, for coming out and your presentation today. It was excellent. Like I said, there's a ton of information there to process. And thank you to everybody else who attended. We appreciate the support and attendance through the series. Craig Willis, you were the last presenter for our series, and we're just letting everyone spool up their busy field season. I think the common plug is, if you like the series, head on over to our YouTube and hit that subscribe button. And I wish everyone good luck over the next few months and look forward to crossing paths with you all in the near future. Thank you again, Craig. It was a great presentation, and we'll let everybody head off now, and I'll stop the recording.
Great. Thanks so much.